Our immune system can produce a vast range of antibodies having different antigen specificities, and these antibodies can recognize the virtually every visual and the potential antigen that we can come across. Yet the millions of the all the antigens are entering inside our body daily, and the other antibodies are also releasing out inside our body, and that they are continuously fighting between them, and the and this increase even the entry of the anything increase the antibody diversity inside our body, and every antibody has a unique antigen binding site, and this is due to the unique amino acid sequence in the variable region, and this amino acid sequence of each protein is encoded by a specific genes, and the human beings can synthesize millions of the unique base antibodies, and every portion is encoded by a particular gene. And so we need millions of genes to encode the huge amount of the antibodies. However, the number of antibodies genes in human body is less than the 200. Now, how this can be possible? There are the millions of genes required, or the only less than 200 is enough for maintaining this diversity. And the question comes. This is the solved by the mystery by the this scientist Tony Guo, um, showing that the antibody diversity arises from the gene rearrangement. What means the gene rearrangement? The gene rearrangement means here the genes for the light chain and heavy chain are present in different chromosomes. Okay, and the genomic DNA, the light chain, in the genomic DNA, the light chain and the heavy chain genes are not complete genes, and the genes are then split into the segments, and these segments are brought together when a new B cell is being formed. And when here the light chain gene rearrangement, how this take place, the light chain gene is made up of the three segments: variable segment, joining segment, and the constant segment. There is the VJC. VJC recombination take place in the mm, light chain gene rearrangement. Here the genes for the V, J, and C segments are present in the three different clusters of the two chromosomes, and the gene for the kappa light chains are present on the chromosome 2, whereas the genes for the lambda um, light chain are present on the chromosome 22. For the kappa light chain, there are 40 genes for the B cluster, whereas 5 genes in J cluster, and the 40 in these 1 genes in the C cluster. Whereas for the lambda light chain, there are 30 genes in B cluster, 4 genes in J cluster, and 4 genes in the C cluster. And the V segment and the J segment gene encode the variable region of light chain, and the C segment gene encodes the constant region of the light chain. These the variable region are encoded by the V segment and the J segment, and here the C segment is encoding the light chain. When a B cell differentiates, one of the B segment gene join one of the J segment gene. Okay, the intervening DNA is deleted, and this is known as VJ joining. Similarly, this VJ joining and the C segment gene are transcribed to form the hnRNA. This hnRNA is spliced to form a messenger RNA for a light chain. And here we can see these are the V and the J chains, V and the J regions. When these V and J are joining, the gene rearrangement took place, and this can be happened to the anything. Here we can see the V2 is joining with the VJ, J5. Here the up to V40 is there. So the many combination can be possible, and the J having the 1 to 5, and these combined with the 1 can combine with the 2, or combined with the 40, there is a huge. And these after that, the transcription and splicing take place, and the light chain messenger RNA is formed, and after that, this new antibodies are generated. Here there are 40 genes for the V segments. I've already seen that, and the 5 genes for the J segment of the couple light chain. Therefore, 40 into 5, that is 200 different VJ combinations are possible. There are 30 genes for V segment and 4 genes for the J segment for the lambda light chain. Hence, the 40 into 4, that is 120 VJ combination are possible. And the each a given V cells have a one particular V or the VJ combination. Then the in the this heavy chain gene rearrangement, here the other genes are also involved. A heavy chain gene is made up of the four segments, basically the variable segment, diversity segment, joining segment, and the constant segment. VDJC. These are the VDJC. Here the heavy chain gene are present in the four different clusters on the chromosome 14, and VDJ segment encode the variable region, whereas the C segment encodes the constant region. For the heavy chain, and there are 40 genes in the V cluster, 25 genes in the D cluster, and 6 genes in the J cluster, and 9 genes in the C cluster. And when a B cell differentiate, one D segment one D segment gene join one J segment gene, and the intervening DNA is deleted out, and this is known as the DJ joining. Whereas one B segment gene joining the DJ combination, the intervening DNA is deleted. This is known as the VDJ joining. And VDJ joining and joining combination and the first C segment gene are transcribed to form the hnRNA. This hnRNA is then also spliced out to form a heavy chain messenger RNA. This the VDJ portion encodes the variable region, and possibly the VDJ combination are 40 into 25 into 6, around 6,000, and a given B cell has only one particular combination. And here we have the heavy chain gene rearrangement, the V having the 40 V segments, and the 25 D segments, and the 6 J segments. They are combining and forming the VDJ regions, and these are transcribed out and forms the messenger RNA containing the different combinations of these VDJ. And thus the several different light chains can be formed, and each has having the unique amino acid sequence in its variable region. And several thousand different heavy chains can be also be formed, and each having the unique amino acid sequence in its variable region. And when this light chain combined with the heavy chain, a million of combinations are also possible, and each combination is specific for the one antigen. So this is the diversity, how this diversity is maintaining inside the antibodies. A given given B cells has only one combination, and it synthesizes only one specific antibody. And this combinatorial diversity, in this the variable combination of differential gene segments in the major mechanism of the antibody diversity, and the antibody diversity thus generated is known as the combinatorial diversity. Nearly the 2 million variable regions can be formed by this combinatorial diversity. And the gene rearrangement reaction are catalyzed by some of the enzymes such as the DNA repair enzymes and the RAG1 and the RAG2. And these RAG1 and RAG2 are encoded by the recombination activating genes 1 and the recombination activating genes 2. And the junctional diversity. A second source of the diversity is the junctional diversity, and when the different segment join, some nucleotides may be added or get deleted from the end, and this creates a new base sequence. And these nucleotides are added by the terminal deoxyribonucleotide transferase, that is adding these TDT are adding the nucleotides, and for deletion we having the enzyme exonucleases that can delete out these nucleotides from the end, and the somatic hypermutation. And the final source of the diversity is the somatic hypermutation. When a mature B cell encounters an antigen, a series of point mutations can occur in a variable region of a gene, and this results in the further diversity. Then, yes, there is the huge diversity and the rearranging arranged heavy chain genes. Like the other genes, the heavy chain genes are preceded by a promoter. Uh, a regulator is present upstream of the promoter. An enhancer element is present between VDJ and the C segment. There are the 9C constant segments, and each C segment is preceded by a short repetitive sequence known as switch sequence. And the promoter is weak, but the gene expression is increased by the enhancer element. Here we can see this as a regulator, promoter, variable regions, enhancers, the switch sequences, and the constant region for the heavy chain genes. And the constant, there is the mu, delta, epsilon, alpha, and they are different C segment for the heavy chain genes. Among the 9C segments, the first is the mu C. After VDJ joining, VDJ is transcribed with the first constant segment, that is mu C. And IgM is the first immunoglobulin to be formed upon entry of the antigen. This is the reason why the immunoglobulin and IgM is first synthesized in our body for the and for any of the exposure with the dose of the antigens inside.